Hey guys, Assalamu alaikum everyone. I hope you all are doing well. In the last video, we did till here of biological molecules, right? If you haven't seen part one of biological molecules, please go and check that out. This is part two, and we are starting with the comparison of characteristics of carbohydrates. So, in the last video, we have already studied that there are three basic kinds of carbohydrates. The first one is monosaccharides, then we have oligosaccharides and polysaccharides. So the basic difference is clear that monosaccharide contains a single saccharide unit, oligo consists of 2 to 10 saccharide units, and poly contains more than 10 saccharide units, right? Monosaccharides are the simplest carbohydrates and these cannot be hydrolyzed any further, okay? Why? Because they contain of one saccharide unit and we cannot break the saccharide unit, right? And these are highly soluble in water and these are sweetest among all the carbohydrates. These are very sweet and they are also soluble. Soluble why? Because of the simple structure. Okay. Next we move on to oligosaccharides. They are composed of 2 to 10 saccharide units and they have a less complex structure. So they yield at least 2 and maximum of 10 monosaccharides when there is hydrolysis. Okay. If a disaccharide is made up of two monosaccharide units, then it breaks down into two saccharide units. If it is made up of 10 saccharide units, then it breaks down into 10 saccharide units, right? And it is less soluble in water as compared to monosaccharides, but it is more soluble as compared to the polysaccharides, and they are less sweet in taste. Monosaccharides are the sweetest, then we have oligosaccharides, and then we have polysaccharides, which are absolutely tasteless. Okay. So now polysaccharides have got a very high complex structure and they yield at least 11 monosaccharides. Why? Because in oligosaccharides we have from 2 to 10 monosaccharides, right? But when polysaccharides break down, they produce at least 11 monosaccharides. And these are generally insoluble in water, okay? And these are also tasteless. Why are they insoluble? Because of the complex structure. Okay? I hope this is clear. Now we're moving on to the specific topic of monosaccharides, which are the simplest sugars as we just studied. And the chemical formula is CnH2nOn. Okay, which means that hydrogen is in the ratio of 2 to oxygen as well as to carbon. Okay, for example, if I have 6 carbon, my hydrogen will be 12 and then oxygen will also be 6. So hydrogen and oxygen, the ratio of hydrogen and oxygen is 2:1, which is same as the ratio of H2O. Okay. So these are classified by the number of carbons. For example, trioses that contain 3 carbon, pentoses 5 carbon, tetroses which is 4 carbon. Next we have hexoses which is also 6 carbon. Okay, n is equals to 3 to 7. Okay, so basically in one saccharide unit we can have n is equals to 3 to 7, right? Now the functional groups can be aldoses, whether it contains an aldehyde functional group, or ketoses that it contains a ketonic function group. If you studied organic chemistry in your second year or in your MDCAT, you must know what is the aldehyde functional group and what is the ketone functional group. If not, we're going to study this later, okay? Right now. Examples are glucose, fructose, galactose, and ribose. Glucose contains an aldehyde group, so it is an aldose. And fructose, it contains a ketone group, so it is a ketose, okay? Now, the properties of monosaccharides, the first one which we have already studied, that monosaccharides are sweet in taste. These are very easily crystallizable. For example, in glucose and fructose. Wait a second. And these are soluble in water. Okay, why? Because the polar hydroxyl groups, they can dissolve easily in water, but they cannot dissolve in non-polar solvents. Okay, so what happens basically is that water is a polar solvent. It contains H positive and OH negative ions, right? That is why what happens is that it can easily dissolve polar groups, but non-polar solvents cannot be dissolved in this. Okay, now monosaccharides are also reducing sugars. Why? Because they react with the Bendix and Fehling solution. I haven't mentioned this information in the slides because this was I don't know, this wasn't a part of the MDCAT syllabus. It wasn't written appropriately. But if you want more information for this, you can go and study the Federal Board biology book. Okay, it is written, there's a whole table drawn with all the tests of carbohydrates that show how carbohydrate has got a reducing sugar. So basically you just need to remember that it is a reducing sugar. Why? Because they have a free aldehyde and they have a free ketonic group. Now they have a simple structure. They exist in a linear open chain structure, or they can also exist in a ring structure or a cyclic structure, right? And they can be rapidly oxidized to release energy. For example, glucose is rapidly oxidized in the presence of glucose is rapidly oxidized in cellular respiration. Okay, they act as building blocks for disaccharides, sucrose, lactose, and polysaccharides, for example, starch, glycogen, and cellulose. So the basic unit that we need to know, it is glucose, right? So when sucrose is formed, which monosaccharides are needed for the formation of sucrose? We need glucose plus galactose, right? And for lactose, right, we need fructose. Wait. I'm so sorry. For sucrose, we need glucose plus fructose. I'm really sorry. Please correct your concept. But still, wait a second. Yes, guys. So, I just made a terrible mistake. I don't know why. Sucrose is made up of glucose plus fructose, right? And lactose is made up of lactose, which is basically the milk sugar. It is made up of glucose plus galactose. Okay, so please remember these points, right? Next, we move on to the chemical formula and the chemical structure of glucose. Glucose is a hexose monosaccharide. Now, what do you mean by hexose? Hexose means having 6 carbons, okay? And the molecular formula is C6H12O6, right? Glucose is an aldohexose. It contains 6 carbons, which has an aldehyde group at carbon number 1, okay? Aldehyde group is CHO and it is present at carbon number 1. Now, first we're going to study the linear structure of glucose, right? See, this is carbon number 1, okay? And this is the aldehyde group which is present, okay? Right? Now we move on to the cyclic or the ring structure. In aqueous solution, glucose exists in a cyclic form that is known as the pyranose ring due to intramolecular reaction. Okay, so whenever glucose exists in the cyclic form like this in a ring-like structure and it is closed from all the ends, then it is known as what? It is known as the pyranose ring. Okay, the OH group on carbon number 5 reacts with the carbonyl group at carbon number 1 and it forms the hemiacetal bond. Okay, so this OH on carbon number 5, 1, 2, 3, 4, 5, this one. This reacts with carbon number 1 over here and it forms like this ring structure. It encloses itself in a ring-like structure and it forms the hemiacetal bond. Right now, there are two basic possibilities. The first one is alpha glucose, where carbon number 1, where we have difference at carbon number 1, the hydroxyl, it points downwards, right? Whereas in beta glucose, the hydroxyl, it is upwards. Okay, so let us see over here. See, this is the difference which we can see over here, this one. This is alpha D glucose and hydroxyl is at the bottom. This is carbon number 1 and hydroxyl is at the bottom, right? Whereas over here, if you have a look, this is carbon number 1 and hydroxyl is at the top. So this is beta D glucose. Okay, why is it D? Because this is dextrorotatory, which we're going to study it right now. Okay. After a few slides, it's gonna come. Structural isomerism. Glucose has a molecular formula of this, C6H12O6, but its atoms can be arranged differently. For example, glucose is an aldohexose, right? Its aldehyde group is at carbon number 1, and fructose is a ketohexose, which has a ketonic group, okay, at carbon number 2, right? So both of these have got a similar structure. Both of them have got the same number of carbon atoms, the same structure, but there's a difference of functional group. So this is functional group isomerism. The glucose has got an aldehyde group, which is CHO, and the fructose has got a ketonic group, which is C double bond O, okay? So this you must have studied in your organic chemistry. Both of them are structural isomers, which means that there is a different functional group, but they have same molecular formula. The molecular formula is same. They have got the same number of atoms. They have got the same everything structure as well, but they have different functional groups. Within hexoses, glucose is an aldose, while fructose is a ketose. And they have the same molecular formula, but they have got different functional groups. So you need to remember this, that this is an example of structural isomerism. Why? Because the functional group has been altered. You see over here, this is carbon number 1. And on carbon number 1 in glucose, we have what group? We have got aldehyde group. Aldehyde group. And over here on carbon number 2 in fructose, we have got a ketone group, right? Now we move on to stereoisomerism in glucose, right? Stereoisomers, it means that same molecular formula and structure, but they have different spatial arrangement of the atoms. It means that the structure is the same, the molecular formula is all the same, everything is the same, but only the arrangement of the atoms in space, it is only different, okay? Now we move on to enantiomers. This is a kind of stereoisomerism and it depends upon the arrangement of hydroxyl group on the penultimate carbon. What is penultimate carbon? For example, if we say pentagon, so it means five-sided, right? Similarly, if we say penultimate, it means at which carbon? At carbon number 5 of glucose, right? D-glucose hydroxyl group OH is on the right. Naturally occurring form, and this is biologically active. L-glucose OH is on the left, rare, not metabolized by humans. So the difference between both of them is, as you look over here, this one is D-glucose. D means dextrorotatory and L means levorotatory. Right? So when I say dextrorotatory, it means that my OH group is on the right hand side, right? And when I say levorotatory, L for levo, L for left, okay? So it means that my hydroxyl group is on the left hand side, right? Now this D-glucose, it is very common in humans. This is the most common form. L is rare and it is rarely metabolized. It cannot be metabolized in humans, okay? So basically, enantiomers are what? Enantiomers are mirror image stereoisomers, but these are not superimposable over one another. As you can see over here, if I keep this surface in between, consider this to be a mirror, right? So they form a mirror-like image of each other. Why? Because these are totally symmetrical like this, okay? So that is the scene, right? I hope it is clear. Now we move on to the diastromers. They are not mirror images of one another and they differ at one or more chiral centers. Okay, so they can differ at one or more points and these are not mirror images. If enantiomers are there, so these are mirror images and their examples are D-glucose as well as L-glucose. Okay. Yep, it's recording. I thought it wasn't recording. So the examples are D-glucose versus D-mannose. These differ at carbon number 2. Let's have a look over here. D-glucose, dextrorotatory glucose and dextrorotatory mannose. They differ where? They differ at carbon number 2. Okay. Over here, if you see, for example, this is the right hand side, this is the left hand side, right hand side and left hand side. And if you compare all of these hydroxyl and OH and H group arrangement. So all of them are, symmetrical to one another and all of them are, sorry, not symmetrical, all of them are arranged in a similar way. For example, have a look over here. This point is same, okay? Then I have a look at this carbon. H is on the left. Over here, H is on the left as well. OH is on the right. Over here, OH is on the right as well. Next, have a look at this carbon. H is on the left. Over here as well, then OH on the right, then over here OH on the left and H on the right. Similarly over here, but it differs at this carbon, this one. Okay, let me just highlight this one. Okay, so what happens over here? On the left hand side we have got hydrogen, but on the right, on the left hand side in mannose we have got OH group. So this is the difference at carbon number 2, and this is known as diastromers. Glucose and mannose are diastromers. Why? Because the arrangement is not the same at carbon number 2, right? Now, similarly, we have got the D-glucose versus D-galactose. They differ at carbon number 4. You need to remember the carbon number at which the difference is occurring. Okay. Now, D-glucose and D-galactose, they differ at carbon number 4. At this carbon. Okay, so you see the difference, what's happening over here. On the left hand side we have hydrogen, and on the right hand side we have OH. Whereas over here, if I mark L and R left and right, on the left hand side we have hydroxyl, whereas on the right hand side we have hydrogen. So this is a major difference over here. That is why these are also, these are also diastromers of each other. Okay, so there are two diastromers of glucose. The first one is mannose, and the second one you see is galactose. Okay, now let's move on to the next page. I hope this is clear. If not, please let me know. Epimers. This is a special subtype of diastromers. They differ only at one chiral carbon. For example, glucose versus mannose, these are epimers at C2, and glucose versus galactose, these are epimers at C4. Okay, so epimers is what? Diastromers, basically they can differ at more than one carbon. For example, if I say it is differing at carbon number 2, 3, 4, then it is known as a diastromer. But if it is differing only at one carbon, then it is known as what? It is known as an epimer. Okay, if it is a mirror image of one another, then it is known as an enantiomer. So you need to remember all of the differences, okay, in between the terminologies. These are very important. So glucose versus galactose, these are epimers at C4, and glucose versus mannose, since they also differ at only one position of carbon, these are epimers at C2. Epimers are basically the diastromers which differ at only one carbon. Okay, so when I say that all epimers are diastromers, is this statement true? Yes, this is true, because epimers are just diastromers which differ at one carbon atom. But if I say that all diastromers are epimers, is this statement true? No, this statement is false. Why? Because diastromers can differ at more than one carbon atom, whereas epimers differ only at a single carbon atom. So I hope this is clear. Next, we move on to alpha and beta form of glucose, which are known as anomers, which we just studied over here. Let me just show them to you. Over here, when we are studying about the ring structure. Okay, so the difference in the rings, the difference of the alpha and beta glucose, these structure are basically known as the anomers. Okay, let's have a look over here. The orientation of the hydroxyl at the C1, which is the anomeric carbon, it differs. Okay, in alpha glucose, it points down, trans to the CH2OH group, and in beta glucose, it points up, which is cis to the CH2OH group. Okay, see. Over here, this is trans to the CH2OH group, and over here, this is cis, okay, to this group, right? And what the basic difference is just that in alpha, this is gonna be downwards, and in beta, this is gonna be upwards. The OH group is gonna be upwards, right? Now, the difference is crucial. In alpha glucose, we have got these examples, starch and glycogen, and in beta glucose, we have got cellulose, right? So, whenever the carbon is downwards, right? Sorry, whenever the hydroxyl group is downwards of carbon, it means that the structures which can be formed are starch and glycogen. Cellulose cannot be formed, okay, because cellulose is a form of beta glucose. Beta means that hydroxyl group will be up. Okay, so whenever the hydroxyl group will be up, it can never be starch and glycogen, it will always be a cellulose. So they can give you a simple structure, and they can say that this is the structure, and the hydroxyl group is up. Now tell which molecule can this be, starch, glycogen, or cellulose. So you need to identify according to this position of the hydroxyl group that which can be the answer. Okay. Cyclization of glucose, it produces new asymmetric center at C1. The two stereoisomers, these are known as anomers, and Howard projections represent the cyclic sugars having essential planar rings with hydroxyl at the anomeric carbon 1. These are known as Howard projections. You don't need to remember this, it's just written, and this is just the summary that alpha is OH below the ring, beta is OH above the ring. Okay. Now we move on to oligosaccharides. What is oligosaccharides? Basic difference is that mono was with one saccharide unit, and oligo means two or 2 to 10 saccharide units which are linked together by the glycosidic bonds. Okay, the most common are disaccharides. The first one is sucrose, cane sugar, which is composed of glucose and fructose, and it has got a beta 1, 2 glycosidic bond. Okay, now how is this glycosidic bond formed? As you see over here, this is glucose and this is fructose, right? So over here, this is carbon number 1 of glucose and this is carbon number 2 of fructose, right? Now what happens over here, basically on carbon number 1 of glucose, what happens is that this OH and this OH, we have got these two OH. These two will do a reaction together. What will they do? Basically, this OH will be eliminated. Okay? Eliminated. And over here, from over here, what will we take? We will take H positive only. Okay? And this oxygen will form a bond with this hydrogen at the top. Right? And this one, this H and OH negative will form water together. Okay? As you see over here, right? First we had OH over here and we also had OH over here, right? Now from here only H is eliminated and this O, oxygen, goes and forms a bond with this hydrogen, okay, of carbon number 1 of glucose, and this hydroxyl is removed completely and it forms a bond with hydrogen. In order to form what? It forms water. So it forms alpha 1 comma 2 glycosidic linkage. Why alpha? Because the this one below, the hydroxyl group which is below, it is removed. Okay, that is why alpha 1 comma 2. Why? Because one carbon number 1 from glucose and carbon number 2 of fructose. Okay, these two do a reaction and in between these two carbons what forms? A glycosidic linkage is formed. Okay, so whenever this sort of a structure is seen in between two monosaccharides, what will you say? That this is the glycosidic linkage. Okay. The sources are cane sugar and beet sugar. It is a non-reducing sugar. Why? Because no free hydroxyl group at the anomeric carbon. Okay. There at the anomeric carbon, if we see, there is no free hydroxyl group. Okay. All of the hydroxyl groups are occupied together. Right? So that is why it is a non-reducing sugar. So you need to remember this point that they ask you in MCQ that which of the following is a non-reducing sugar. So the answer would be sucrose. This is a major form of sugar in plants, and this is a table sugar for humans. So the sugar which we eat, it is made up of glucose and fructose, which together form what? They form sucrose. Okay, and it is a form of the transport of sugar in plants as well. Now we move on to the lactose. What is lactose? Lactose is glucose plus galactose. Okay, and beta 1 comma 4 glycosidic bond. See. As we look over here, what is happening over here is that, see, this is the H, this is the H as well. Okay. Now what happens over here is that this one, basically what happens over here is that this one removes, OH negative removes, right? And from here also hydrogen is removed, right? And OH negative plus H positive, what does this form? This forms H2O, water, right? And this O, it forms a linkage with this carbon, this hydrogen, sorry, okay? The oxygen of galactose, it forms a linkage with the hydrogen of glucose, okay? And this occurs at carbon number 1 of galactose and carbon number 4 of glucose. That is why this is known as beta 1 comma 4 glycosidic linkage. Okay, so this is why you must know how this is formed now. This is known as the glycosidic linkage, and it leads to what? It leads to the formation of water. Okay. You need to remember the glycosidic bond, which is present in all of these. Sucrose has got alpha 1, comma 2 glycosidic bond, and lactose has got a beta 1, comma 4 glycosidic bond. Lactose is a milk sugar, that is why it is formed in milk, right? And this is a reducing sugar. Free hydroxyl is present at the glucose end. Okay. There's a free hydroxyl group which is present over here, whereas in sucrose we don't have a free hydroxyl group. That is why this is non-reducing sugar, and sucrose is the only sugar which is non-reducing and included in your MDCAT syllabus. So please remember this as a differential point. Okay. The importance is that it is a main carbohydrate in mammalian milk, and intolerance occurs in lactose deficiency. When a person is lactose intolerant or lactose deficient, what happens is that he cannot digest lactose. Okay. So he cannot then consume milk. Now we move on to maltose. Maltose is what? Maltose is glucose plus glucose, and it has got alpha 1, comma 4 glycosidic bonds, which is formed during starch digestion. What happens over here when starch digestion takes place in our mouth when we eat, for example, I ate bread for breakfast, right? Now what happens to the bread? This starch, it will break down into maltose. How will it break down into maltose? Basically, there are enzymes. Enzyme is known as amylase. And since it is in my saliva, so it is known as the salivary and amylase. Okay. And then what happens is that this starch is converted into maltose. And this is an intermediate in carbohydrate metabolism. Okay. Right? Starch is converted into maltose, and this is an intermediate. Why? Because now maltose has to convert into simplest forms, which is glucose. Okay, that is why. Now, as you see over here, this is a glycosidic linkage being formed. What happens over here? There is one H positive and OH negative, which forms what? Which forms H2O, and this is water. Okay. Orientation of this OH group identifies these groups as disaccharides as alpha maltose. Why? Because see, this is down. OH group is down. So it means that this is alpha. Okay. If it was up, it means that it is beta, right? So this is how the glycosidic linkage is being formed over here, and this is the formation of water, and it is 1 comma 4. Why? Because one carbon number 1 of alpha glucose plus carbon number 4 of alpha glucose, these form what? These form a glycosidic bond. Next we move on to polysaccharides. Polysaccharides are long chains of monosaccharides, mainly glucose units, which are linked together by glycosidic bonds, and they serve as storage molecules. They provide a very strong and structural support. Okay, now we move on to the first polysaccharide, that is known as starch. Starch is found in potato, rice, wheat, and corn, and it is made up of amylose and amylopectin. Okay, the first one is amylose, and the second one is what? We have got amylopectin. Amylose is unbranched, it has got 1 comma 4 bonds, alpha 1 comma 4 bonds, whereas amylopectin, it is branched. Okay, they are gonna ask you the structure, and it also has got two kinds of bonds, which are 1 comma 4 and 1 comma 6 bonds. Okay, and it is a major form of glucose in plants. It acts as a primary energy source for humans, right? So this is amylose, and this one is amylopectin, right? Amylose only has got 1 comma 4 glycosidic linkage. This is carbon number 1, this is carbon number 4, right? And amylopectin, it has also a branch linkage, okay, which is 1 comma 6 branch linkage. It has got 1 comma 4 linkages, which are these ones, okay, and after that it also has 1 comma 6 linkages, which are these diagonal ones. Okay, so this is how amylose and amylopectin is formed, and starch is formed through these amylose and amylopectin. All right, now we move on to glycogen. Glycogen is stored in the liver as well as in the muscle cells, and it is highly branched. It has got alpha 1 comma 4 and alpha 1 comma 6 bonds. Okay, and it maintains the blood glucose levels. It provides lots of energy. It is known as the animal starch. This is very easy. It is more branched as compared to amylopectin. Amylopectin is present in plants, right? Whereas glycogen is present in animals, which is even more branched. Okay, and it has got alpha 1 comma 4 and alpha 1 comma 6 bonds. Okay, as you see over here, these bonds plus these bonds, it is diagonally also, joining these other carbons. So this is, branched just like a tree, right? Now we have cellulose. Cellulose is a plant polysaccharide. It is found in plant cell wall, as you know, and this is linear. This is unbranched chain of glucose with beta 1 comma 4 bonds. This is not branched together. Okay, whenever there are two linkages, it means that the structure is branched. Whenever there is one linkage, it means that the structure is unbranched. It provides rigidity and strength to the plant cell walls, and it is not digestible by humans. Okay, there is no cellulase enzyme, and it acts as fiber. Okay, because humans do not have cellulase enzymes, that is why it is not digested by humans. And these are beta 1 comma 4 glycosidic linkage. This one, okay, this is unbranched, right? So we are done with the topic of carbohydrates. And from proteins, I'm gonna start a new video in which we are gonna discuss proteins, fats, and what else? We have nucleotides as well. All right, so I'm ending the video over here now. Inshallah, let's meet in the next part because this chapter is really, really long. So I wanted to explain it thoroughly to you guys. And if you have any queries, any suggestions, please let me know. Till then, take care and Allah Hafiz.